Hello everyone and welcome to this explainer. Today we are diving into a truly dramatic story straight from the world of biotechnology. We're talking about the groundbreaking pioneer of next-generation sequencing, a legendary piece of tech known as 454 sequencing. Now, this system completely changed the game in genomics. It literally opened the door for massive parallel DNA analysis. But despite its absolute brilliance, it tragically didn't survive the highly competitive market that it essentially helped create.
So to understand both its incredible success and its ultimate downfall, we have to start with this absolutely fascinating question. How exactly did scientists sequence millions of DNA strands by turning them into microscopic flashes of light? I mean, it sounds like straight-up science fiction, right? But it was very real biochemistry. It's the brilliant mystery of a now defunct technology, and today we're going to solve it together.
All right, here is our roadmap. We've got 6 parts. 1, the first next-gen sequencer. 2, preparing the DNA library. 3, one bead, one DNA. 4, translating DNA into light. 5, the homopolymer fatal flaw. And wrapping up with 6, 454 versus the gold standard. Let's get right into it. Section 1, the first next-gen sequencer, a pioneer's timeline. This is the definitive rise and fall story. It begins back in 2003 when a company called 454 Life Sciences developed the very first next-generation sequencing, or NGS, platform. And it was a massive breakthrough. It was so big, in fact, that it led to a huge acquisition by the pharmaceutical giant Roche in 2007. But just 6 years later, in 2013, it was completely discontinued. Basically, it was outcompeted by cheaper, higher-throughput platforms like Illumina. But to really grasp why 454 was such a game-changer back in 2003, we need to quickly lock down a couple of basic terms.
Let's start with reads. A read is simply a short DNA sequence physically output by the sequencer itself. Now, the length of these reads varies by platform, but what made 454 so special at the time was that it produced average reads of 400 to 600 base pairs, and sometimes even up to 1,000. Compared to early competitors who were only putting out, 35 to 50 base pair reads, 454 was giving scientists these massive long strings of genetic text, and longer reads give you way more genomic context, which makes computationally assembling and mapping a whole genome significantly easier.
Another key concept to keep in mind is sequencing depth, which is also known as coverage. This is just a statistical measure of how many times a specific nucleotide in the genome has been sequenced across all the reads in your run. So for example, 30x coverage means every single base in the genome was read an average of 30 times. This is absolutely vital for accuracy. You want to read the same base multiple times to be 100% sure your sequencer didn't just make a random mistake. Think of it like proofreading a paper a few times to catch typos.
Okay, Section 2, preparing the DNA library. Setting the stage.
The prep phase to get the DNA ready follows 4 pretty straightforward steps. First, fragmentation. We take that long, bulky genomic DNA and break it down into manageable chunks, about 300 to 700 base pairs for the 454 system. Second is adapter ligation. We tack on specific chemical sequences to the ends of these fragments, which the machine literally needs so it can grab onto and amplify them. Third up is amplification. We make a ton of copies of the fragment to boost the signal intensity for later. And finally, step 4, the actual sequencing.
Now, during that adapter ligation step we just mentioned, scientists do something incredibly clever. So the crucial point here is this concept called multiplexing. On the 3 end of the adapter, scientists can attach a unique 8 to 10 base pair identifier. It's essentially a literal genetic barcode. What does this mean? It means you can take 96 completely different samples and just mix them all together into a single sequencing run. The machine reads the whole mixed batch at once, and afterwards you computationally sort the reads by their unique barcodes back into individual samples. It is a massive time saver, and it dramatically reduces the per sample cost, which is what made large-scale genomic research actually accessible to everyday labs.
Moving on to Section 3, one bead, one DNA, the emulsion PCR miracle. So, to actually sequence the DNA, we need a really strong signal, which means we need to amplify it. But we absolutely cannot let different DNA sequences mix together, otherwise we'll just get a jumbled, unreadable signal. The genius solution to this? Emulsion PCR. The fragmented DNA is mixed with these tiny microscopic beads. The goal is to have exactly one DNA fragment bind to exactly one bead. They are then mixed into a water-in-oil emulsion, basically like shaking up vinaigrette dressing. This creates millions of microscopic water droplets entirely surrounded by oil.
Each little droplet acts as an isolated microreactor containing just one bead, one DNA fragment, and the chemical reagents needed for PCR amplification. Inside that tiny, isolated water droplet, the PCR reaction takes off, amplifying that single DNA fragment over and over and over again until it completely coats the surface of the bead. The result? You get roughly 1 million identical copies of a single DNA molecule attached to one single microscopic bead. And because it's completely trapped inside that oil-coated droplet, there is zero cross-contamination from any of the other DNA fragments.
Now, what is truly mind-blowing here is the scale of this operation. Once those beads are fully coated in DNA, they're deposited into what's called a picotiter plate. A picotiter is 10 to the negative 12th liters. I mean, that is an unfathomably small volume. We're talking barely anything. Each well on this microscopic plate holds exactly one bead, along with the specific enzymes needed for sequencing. This insane design allows hundreds of thousands of individual sequencing reactions to happen simultaneously, in parallel. That's what gave 454 its massive throughput.
Section 4. Translating DNA into light. The magic of pyrosequencing.
Okay, so pyrosequencing works through a brilliantly elegant four-step biochemical pathway. Think of it like a molecular relay race. First, a DNA building block, a DNTP nucleotide, is added to the well. Second, if that nucleotide happens to be a match for the sequence, the enzyme DNA polymerase incorporates it and spits out a byproduct called pyrophosphate, or PPI. Third, a second enzyme called ATP sulfurylase grabs that PPI and turns it into ATP, which is basic cellular energy. And finally, step 4, the grand finale, an enzyme called luciferase uses that newly created ATP to emit a literal flash of visible light. A supersensitive photomultiplier tube detects that flash. So, in real time, the invisible DNA letter has literally just been converted into a visible photon.
But wait, how does the machine actually know which specific letter was added? Well, it does this through something called stepwise DNTP addition. Basically, the machine washes the plate and adds only one type of nucleotide at a time. Let's say it just washes the plate with A's. If the sequence needs an A, the reaction happens and the well flashes with light. If it doesn't need an A, nothing happens, it stays totally dark. The machine then washes the A away and tries again with C. So base identification is literally just a matter of the computer tracking which chemical cycle produced the light. Simple, but genius. Let's look at Section 5, the homopolymer fatal flaw, the tragic downfall.
So the villain causing the downfall of this amazing system is something called a homopolymer. This is simply a sequence of identical DNA bases in a row. Think of a string of 7 A's or 6 T's all lined up. Now take a second to really think about the light flashing mechanism we just talked about. Why would a simple repeating sequence of identical letters break this brilliant million-dollar system?
Well, here is the underlying physics problem. If the machine washes A over the plate, and there are 2 A's in a row in the biological sequence, 2 A's get incorporated, right? So it emits twice as much light. 3 A's emit 3 times the light. All good so far. But when you hit 6 to 8 bases or more, the light signal no longer scales linearly. The optical detector simply cannot distinguish the brightness of 6 photons from 7 photons from 8 photons. It's just a blurry, bright flash. The result is a highly systematic insertion or deletion error rate, which we call indels. The machine literally just loses count. And in genetics, losing count causes massive frame shifts in coding regions.
To give you a concrete example, if the true biological sequence has 8 A's in a row, the 454 sequencer might easily read it as 7 A's, simply because the flash of light for 7 looks basically identical to the flash of light for 8. And here's why that's catastrophic. A single missed letter shifts the entire downstream reading frame of the DNA. That means scientists might computationally predict a completely non-functional protein entirely by mistake. For complex genomes with lots of repeating letters, this was a massive, massive limitation.
And compounding this accuracy problem were the astronomical costs. 454 relied on very, very expensive enzymes like luciferase and ATP sulfurylase. Plus, it required time-consuming wash cycles to clear out those nucleotides one by one. By 2010, running 454 cost around $500 to $1,000 per million reads. Meanwhile, nimble competitors like Illumina were aggressively dropping their prices down to just $10 to $50 per million reads. 454 was simply too expensive and way too error-prone with those homopolymers to survive the market. Which brings us to our final section, Section 6, 454 versus the gold standard, the legacy.
When we compare these platforms side by side, it becomes painfully obvious why 454 met its end. Sanger sequencing, which is actually a much older technology, produces very long, highly reliable reads of 800 to 1,000 base pairs. More importantly, while Sanger has a 99.9% accuracy rate, any errors it does make are entirely random. 454's errors, on the other hand, were systematic. It would reliably and predictably fail every single time it hit a long homopolymer. Because of this, Sanger sequencing remains in use today as the undisputed gold standard for verifying sequences, while the brilliant, light-flashing 454 system was permanently discontinued in 2013.
Which leaves us with this final thought. Look, 454 sequencing was an absolute masterpiece of biochemistry. It turned invisible genetic code into microscopic flashes of light and effectively ushered in the modern era of next-gen sequencing. But it hit a hard physical limit in how light scales. So as we look at today's bleeding-edge genomic technologies, we really have to ask ourselves, what brilliant, invisible limits will our next generation of tech need to overcome? Keep that in mind as you continue exploring the ever-evolving world of biotechnology. Thanks for joining me for this explainer, and keep asking those tough questions.